So for today's video, I will discuss about biochemistry of nucleic acids. When we say nucleic acids, it includes the two processes which called the DNA and RNA to carry genetic information which is read in cells to make RNA and proteins by which living things functions. But the well-known structures of DNA is only a double helix which allows the information to be copied and passed to the next generation. So for nucleic acids, they have two main classes which are deoxyribonucleic acids, which better known as DNA, and ribonucleic acids, which better known as RNA. These molecules are composed of long strands of nucleotides, which held together by a covalent band. Nucleic acid can be found within the nucleus and cytoplasms of cells. So nucleic acids are biopolymers or small biomolecules which are essential to all known forms of life. They are composed of nucleotides which are monomers made of three components which are 5-carbon sugar, a phosphate group, and a nitrogenous base, and if the sugar compound is ribose, the polymer is RNA, and if the sugar derived ribose as deoxyribose, the polymer is DNA. So nucleic acids are the most important of all biomolecules. They are found in abundance of all living things, which function to create and encode and then stored information in the nucleus of every living cells of every life form organisms on Earth. It also functions to transmit and express that information inside and outside to the cell nucleus, to the interior operations of the cell, to the next generation of its living organisms. The encoded information is contained and conveyed to the nucleic acid sequence, which provide the latter step ordering of nucleotides within the molecules of RNA and DNA. Nucleic acid which store, transmit, and help to express hereditary information. The amino acid sequence of a polypeptide is programmed by a unit of inheritance which called genes. Genes are made up of DNA, and nucleic acids are made up of monomers are called nucleotides. So there are two types of nucleic acids, which are the DNA and RNA. DNA provides direction for its own replication, and it directs synthesis of messenger RNA or mRNA, and mRNA controls protein synthesis and which occur on ribosomes. So nucleic acid is made up of monomers called nucleotides. Each nucleotide consists of nitrogenous base like pentose, sugar, and one or more phosphate groups. So there are two families of nitrogenous bases, which are the pyrimidines. It contains of three elements of sugar, which are the cytosine, thymine, and uracil, which have a single six-membered ring, while purine, it contains of two elements of sugar, which are the adenine and guanine, which have a six-membered ring fused to a five-membered ring. In DNA, the sugar is deoxyribose, while in RNA, the sugar is ribose 15. So RNA molecules usually exist as single polypeptide chain, while DNA molecule have two polynucleotides around imaginary axis to form a double helix. In the DNA double helix, the two backbones run in an opposite direction from each other, and one DNA molecule includes many genes. So the nitrogenous bases in DNA pair up and form a hydrogen bond, with adenine always with thymine and guanine always with cytosine, which is called complementary base pairing. Complementary pairing can also occur between two RNA molecules or between part of the same molecule in RNA, and thymine is always replaced by uracil. Among the two purines, guanine and adenine, and the three pyrimidines, cytosine, thymine, and uracil, thymine is found in DNA and it replaced by uracils in RNA. Both thymine and uracil can form base pair with adenine. DNA and RNA are polynucleotides. Two of the phosphate are lost when the base is polymerized into a polynucleotide. The remaining phosphate is part of the main chain. The phosphodiester backbone is comprised of deoxyribose, DNA or ribose in RNA, sugars, biphosphates between the 3 and 5 positions of the sugars. The polymers are formed in nucleic acids. The 3 oxygen attacks the 5-phosphate of nucleus or nucleotides triphosphate, which release phosphate pyrophosphate and forming a phosphodiester linkage between the two nucleosides. The important functions of nucleic acids are the genetic information is coded inside DNA. This information is stored by the DNA and transmitted to the proteins by RNA. The mRNA migrates from the nucleus to the cytoplasm to form the foundations for protein. And the tRNA makes use of the amino acids in the pool to arrange the protein's information. The rRNA is useful for making up ribosomes. And remember that the main functions of nucleic acid is to store information, which is for DNA, protein synthesis, which is for RNA, and energy transfers, which is for adenosine triphosphate or ATP, and nicotinamide adenine dinucleotide or NAD.